starting hypersensitivities type 1, 2, 3, 4, which are respectively immediate, subacute, intermediate, 3, and then finally number 4 is delayed. I'm looking here at the immediate hypersensitivities, and they're usually initiated with allergens of some kind or another. They could be allergens related to pollen, to dust, to any kind of protein aerosol, as it were, and there could be some other items out there like danders. Basically, we're going to start an immune response. The immune response, which is this situation in which the macrophage presents something to a T helper cell, which activates a B cell, which activates plasma cells, in this case, to make immunoglobulin E. And there could be a more acute version of the type 1 immediate, which is anaphylaxis. I have a handout for you at this time, and looking at it in the landscape mode here, we have type 1 hypersensitivity here. Duration is seconds, or rather onset is seconds, and duration is about 20 minutes if there's no further excitation or stimulation. And I'm presenting here the idea that it could be pollen, hair danders, hay fever, asthma type things. Sometimes dust will activate asthma. So that's the idea. Particulate matter in the air can do this sort of thing, particularly to asthma that has an underlying inflammatory component. Okay, so first comes the sensitization. We produce the antibodies. Because of the chronic nature, we're going to go all the way to an immunoglobulin E here. And the indication here in terms of class switching is that most of the time we're getting activation with immunoglobulin M and with G. The D is a surface antibody. This is the immunoglobulin G. Gamma globulin is the one that you have persistent in your blood and which crosses the placenta. If you're a mother with milk and you're feeding a baby with mother's milk, it is the immunoglobulin A that's found in the milk. And then ultimately the chronic type of immunoglobulin, which takes time to develop, is the immunoglobulin E. And that's the one that's actually going to seat itself in the basophil here, or mast cell. Right, so we get sensitization. This could be the dust cells out here, or the pollen, or the danders, activating a B cell, which grows up to be a plasma cell, produces antibodies. Here's our antibody, which is the immunoglobulin E, the chronic antibody, and it seated itself tail first into the mast cell. And then with the introduction of the allergens, we're going to get degranulation of the mast cell and the production of histamine, right? There is a more acute version of this, and that's listed for our viewing pleasure on this particular handout, the leukotrienes and prostaglandins. If we have a leukotriene development, we could ultimately get to these type of leukotrienes, which represent the substance of anaphylaxis, type 1 hypersensitivity. These little items right here, which could be actually thousands of times more powerful than histamines. Okay, so that's our type 1 hypersensitivity. Things can get more, how shall I say, intense if, first of all, we go beyond, let's get back to this page, if we go beyond the duration period in which we do not further excite or stimulate. If we go beyond that period due to some sort of excited response or stimulatory response, we could get to a further, more acute episode. The ultimate example of that would be anaphylaxis, which is a system-wide reaction from oftentimes bee stings, penicillin, I spelled that a little bit wrong there, sorry, other drug allergies. There's a wide variety of drugs that will cause allergies in some individuals. And basically the mast cells throughout the body will be activated, resulting in circulatory collapse. So that's everything you need to know about type 1. Type 2 and type 3 are subacute. They both have an onset of 1 to 3 hours, right? They both have a duration of 10 to 15 hours, right? And then we, in the case of type 2 hypersensitivity, this is referred to as the complement-mediated lysis, cell lysis. It's stimulated by IgG and IgM because those particular antibodies have handles which have activation sites for complement. When you activate complement, you get the membrane attack complex, right? And it basically it lyses the cells. The most common case here is red blood cell mismatch when you're getting or giving a transfusion and the blood typing is wrong. And so the activation of then complement results in the lysis of red blood cells. Okay, that's type 2. Type 3, again, subacute, intermediate, 1 to 3 hour onset. People have to live beyond the 10 to 15 hours, and things can get exacerbated. And the issue here is going to be foreign antigens, which can become bloodborne or in the interstitial fluid. And then there could be antigen-antibody complexes. So I should underline that ultimately. Antigen-antibody complexes are what characterize this particular disease state. And I also mentioned to you the lumpy, bumpy quality that you see when you take a tissue sample, mount it on a slide, stain it properly, and then you notice that there's a lot of antigen-antibody complexes that have formed, precipitated out of the fluid, and those stain very dark blue. They take on the basic stain, not the acid stain, right? So they're stained blue, and you just see a lot of lumpy, bumpy types of configurations. And that will occur in the lungs and block the lungs, block the gas exchange of the lungs. It will occur in the kidney, and it will prevent the kidney from filtering the blood, and that's called glomerulonephritis. Okay, why is this ultimately happening? What's the further tissue injury? That's because the neutrophils cannot phagocytize those complexes. They're too large, and so the neutrophils literally explode in what's called a respiratory burst, and they release degradative enzymes and bleach, right? And so they injure the tissues of the lungs or the kidney. Okay, so that's that one right there. And then we have the delayed version. This is the version that involves things like the contact dermatitis, typically of poison oak, poison ivy. A similar kind of contact dermatitis can come from the oxides of metal. Could be lead, could be nickel. And in this particular case, the contact dermatitis, which is a form of rash, a weeping rash, takes about 3 days to develop. So if somebody's out in a park on a Sunday afternoon, they contact poison oak, poison ivy, then by Wednesday, 3 days later, we're going to be having the contact dermatitis. And if it's kept dry, undisturbed, no scratching, etc., this can resolve in as little as 1 day because we actually have cortisol from our own bodies, which are going to resolve this situation. If there's a great deal of disturbance, people decide to bathe or put some sort of ointment on the rash, then it spreads. And there's a continued stimulation as a result, and that increases the duration. Extending beyond 1 or 2 months may result in eczema. This is something that can be seen, and ultimately one has to go and get prednisone shots which themselves can actually create a type 1 hypersensitivity reaction in some individuals. Some people cannot handle the type of activity, the response, their own body's response to prednisone because of the rebound effect. At first it works and people get some relief from their contact dermatitis and the itching, but then there's a rebound once the prednisone wears off. Sometimes their whole body breaks out in hives. So there's some difficult responses when we're using very powerful drugs. Okay, so that concludes our episode with type 1, type 2, 3, and 4 hypersensitivities.